Welcome to this translational research in anterior segment. I'm the one of the two chairs. I'm Wai Kit Chu from Hong Kong. My co-chair, Rohit Shetty, I think he is also engaged in other sections now. So because now the time is 9:45, so we better start now, because I know some speakers, they also have other sections overlapping, so I have to finish a talk and rush to another section to give a talk. So may now we welcome the first speaker, Professor Sain Basu. He's going to talk about corneal regeneration, translational advances with stem cells, hydrogels, and 3D bioprinting. Thank you.
Thank you so much. Can I have a presentation? Good morning to all of you. Thank you so much for making me a part of this session. So I'm going to quickly take you through in the next other mics on. All right, sounds good. Okay, so this talk is going to focus on corneal stromal regeneration and talk a little bit about the work that we are doing on stem cells and also 3D bioprinting and also on hydrogels that are used for the 3D bioprinting. I have the following disclosures. There are a few patents on the products that I'm going to discuss and also the source of funding. So, we work on the ocular surface, and what I'm going to talk to you about is on what is on the right side of the screen, particularly with the corneal stroma. The work that we do that is facilitated by the fact that we have this setup which encourages bench to bedside research at our institute, at the L.V. Prasad Institute in Hyderabad. So we have the basic science labs where you can do all your proof of concept research, finding new targets or new delivery systems or new therapies. And then for translating that, we have the entire setup starting from the 3D printing lab for organ and chip and ex vivo validations, the zebrafish facility, the GMP on site for manufacturing of new drug products, animal labs for preclinical studies, a compounding pharmacy for using products which are already approved for ophthalmic formulations, and then the entire clinical research setup for first-in-human trials, phases 1 to 3. We also are guided by some principles. Just to summarize, what I'm trying to say here is that we want to, of course, understand the regulations and adhere to it because that's for patient safety. But basically, we're trying to find problems that are relevant and that are important, particularly to India and the developing world, and find solutions that are affordable and practical. So we are not trying to find solutions that are impractical, that are difficult to deliver, complicated, or very expensive. So affordability is also something that we have to keep in mind. So the problem that we are focusing on is corneal blindness, and specifically corneal stromal opacification. That's because corneal opacities are the #1 cause of blindness in India and also the rest of the developing world. This is very unlike the West and other parts of the world where endothelial diseases are the #1 cause. And also many of these cases carry not such a good prognosis for penetrating keratoplasty at least. So we have all these alternative strategies that people are working on. You're familiar, of course, with the great work that has been done in the last decade and a half on lamellar corneal surgery. There's a lot of work that's going on keratoprosthesis. But what I'm going to talk to you about today is what is on the right side of your screen, which includes stem cell-based therapy and bioengineering, which tries to also bypass the limitations of what is on your left. So if you think of the corneal stroma and the opacification that we're talking about, which is the pathology that affects the corneal stroma, where it gets deranged and turns opaque. You're mainly talking about the cells which make the extracellular matrix itself, which consists of collagen and proteoglycans, or you think of the corneal stroma as a whole, including both the cells and the extracellular matrix. So in terms of regeneration as well, you can think of regenerating the cornea by using either the cells or the extracellular matrix in isolation or in combination. So before we get into the clinical translation, it's also important to realize that a corneal opacity or scars which cause corneal opacity is not just the opaqueness of the cornea, but also the irregularity and the flattening and the thinning of the cornea. So if you want to restore vision, you have to do more than get rid of the opacity. A great example of this is keratoconus, where you can have a clear cornea, but the patient can't see well because the cornea is very irregular. So just getting rid of the opacity is not enough. And for this, we also have developed these in vivo metrics with which you can, through imaging, quantify the scar so that as you are applying the therapeutics, you know that it is working in the tissue In animal trials, of course, you can take out the cornea and see what is changing, but when you're doing clinical trials, you have to find valid metrics which you can use as endpoints in your clinical trials as well. So first, coming to the cells, here we are talking about the keratinocytes, which are mesenchymal in origin, and this work is based on the seminal work that was done in the Vanderburg lab many years ago, and I worked with Jim on this, and then we are working on translating this technology. So there are cells which are these MSCs, or mesenchymal stem cells, which are located in the peripheral cornea very close to the limbus, which have wound healing properties and are also very, very effective in scar modulation. So they can convert opaque tissue into transparent tissue. So we have worked with this over the last 10 years, and we recently concluded 2 phase 1 clinical trials, which were the first stem cell clinical trials in ophthalmology in India. And these were application of these cells for post-infectious scars, sterile ulcers, chemical burns. And we have now completed the 2-year follow-up on these patients, and we will be submitting the data to the regulatory agencies in India, and hopefully we'll get approval for a multicentric trial. And this is where we would need participation from our partners in India for conducting these. But before you get to clinical trials, there are a few things that need to be sorted out. First is storage and transportation. So if you want to develop a stem cell therapy that does not cost $1 million, you have to think of these things. So one of the clever ideas that we had was to use alginate for preservation and transportation of cell. Using alginate is very useful because you can avoid the cold chain. You can store and transport the cells at room temperature without losing cell viability. And before we went to clinical trials, of course, with our GMP grade mesenchymal stem cells, or HLMSCs as we call them, we also had to demonstrate the safety in preclinical studies and also the efficacy in preclinical studies, both prophylaxis and scarless wound healing, and also in treating established corneal scars. And now we have a way in which we can also produce the cells completely xeno and serum free. So this is what basically we do. These cells are derived from the corneoscleral limbus. So you start with the cadaveric corneoscleral rim, which is from which we extract the limbus, which is cut up into small pieces, digested in the lab, and then cultured in the incubators. Initially, you get a co-culture of MSCs and epithelial cells, but by serial passaging, you can get a pure population of MSCs, which you can then use for transplantation. So this is what I get in the operating room to apply in the patients. And the technique of applying these cells is also very simple. Here you can see that I've just removed the epithelium with a sponge, and the cells are mixed in one of the components of commercially available fibrin glue. So it's a very simple and easy way of delivering the cells, topical application, sandwich between 2 components of the glue which stick to each other. And you can do this procedure under topical anesthesia. If you had a case of burns or if you had an ulcer, you would do it in the same way. You put a bandage contact lens and give the patients prophylactic antibiotics for 1 week. That's it, no steroids, no other medications. So what we have seen in our clinical trials, and we will be releasing the data at the ARVO meeting in May. So if you're interested, you can attend our poster, which details all the clinical outcomes. But what we have seen is that these cells do very well, even for scars, even for formed scars. So this is a post-keratitis chronic scar on your left, and this is the image on the right, which shows how much that has improved, and the surface also becomes more regular. This is a slightly more complicated case of a patient with neurotrophic keratitis and persistent epithelial defect post-Hansen's, and you can see the result post-op. This is just with a single application with no post-operative steroid. So the cells seem to be working very well. We are very excited and moving to multicentric trials. But what we are also doing on the side is working on extracellular matrix, like I talked about at the beginning of our multi-pronged approach. So here, we use corneoscleral buttons that are discarded by the eye bank because they don't meet the optical standards for keratoplasty. These are lyophilized, dried, made into powder, and then converted into hydrogel. The hydrogel itself has wound healing properties which can be used in acute conditions like trauma or inflammation. It's sticky, so you can apply it in lacerations, and it can prevent wounded corneas from developing a scar. And we are also testing it now for use in conditions where inflammation is already present and the scars are already formed. So these are going into clinical trials later this year. And finally, the entire corneal stroma, as I promised. So here we take the help of 3D bioprinting. 3D bioprinting allows us to integrate both the cells and the scaffold. We use the hydrogel that I just talked about as the bio-ink for the 3D printers. So the hydrogel basically is used by the 3D printers to print tissue. So I'll just show you a very short video of how this is done. So on the left, you have an extrusion printer on a mold, which is printing the cornea based on the shape of the mold. And on the right, you have a light-based bioprinter. And here you can see that you can basically customize the shape, the size, the curvature of what you're going to print. And if you come to a lab at LVP, you can get your scans done, and we can print out a contact lens or a cornea, which is basically customized to the shape of your eye. And it looks of this color because it is photocross linked. These are currently in preclinical trials where we are doing rabbit studies where we are implanting and transplanting these 3D printed lenticules on the cornea to assess its safety and efficacy. And we are hopeful that we can go into clinical trials in the next couple of years. What we have now also we are very happy with in terms of the combination that we have in terms of transparency and tensile strength. It's very elusive combination to have a biosynthetic cornea that is both transparent and also has strength. And one of the tests for that is whether it can hold sutures, and that is what we're testing here. So I think the future is very exciting because we already have the cells which are in clinical trials. And I think soon you will have them for treatment of corneal and ocular surface inflammation. We have the hydrogels which are going into clinical trials for corneal wounds and volume enhancement. And finally, when we have the 3D printed lenticules, not only will you have stromal replacement, but you can also use these customized lenticules for even refractive correction or treatment of keratoconus. So these are exciting times ahead, and there are a lot of groups around the world that are working on this. And I'm just happy to present what we are doing. And I must thank my mentor who set me on this path, my great team at LVPI, whose work I get to showcase. Because I have such talented people in my team who do the fantastic work, and also funding agencies, and finally my family from whom I borrowed the time that I'm spending with you. Thank you so much.
Okay, thank you very much. I think now we can have time for one questions. Do you have any questions for the speaker?
Thank you, sir. Which glue you use for the cell to transfer?
This is commercially available fibrin glue. So usually we use Tissile, which is made by Baxter. This is approved for human use and is very commonly used in ophthalmology.
Yeah, I know. Thank you.
And you can use other, like there's a supply chain issue with Tissile, I think, right now, but there are some alternatives that are also available in the India market, which you can use.
Thank you.
Are we expecting cross-linking to be replaced? Cross-linking, has a different application because it prevents progression. But if you are treating keratoconus, the regularity, volume enhancement, that is where it will come in. So you can think of it as an replacement, as an alternative to keratoplasty in advanced keratoconus, or for volume enhancement procedures like CTAC or Bauman's layer transplantation, or even CARES. So you can make, for example, CARES lenticule, you can 3D print them instead of making them manually in the eye bank. With the trifines that are the way that is done now. Thank you.
Thank you very much. Yes. Our next speaker, Professor Pooja Karma. Yes. She's going to talk about the next topic, from markers to maps, imaging and biomarkers in personalized eye health. Thank you very much.
Thank you, sir.
Yes,
thank you, sir, and thank you, APAO and the entire Translational Research Committee for giving me this opportunity to share our work, where I'm going to talk about markers to mapping a road to personalized medicine. So there are various diseases we come across and deal with every day, but one of the most common diseases is dry eye, which is multifactorial, and it can impact the quality of life. And at this point of time, an average of 60% of the patients across the world do suffer from dry eye in a mild, moderate, or severe way. Now, we do come across these patients every now and there, where the first set of patients, when we treat them based on your Schirmer's, T-BUT, and tear film metrics, they respond to the treatment, and they're quite happy. However, the second group of patients, they're not happy with the first line of management, and then they start seeking opinions from different doctors. And after multiple opinions and different treatment regimens, they're still unhappy with what they are doing or what treatment they're getting. So we are moving towards a world where we see quite significantly a lot of non-responders in our practice to the initial treatment, where they may have persistent or worsening symptoms with the initial treatment, and that's when they don't respond. So what we do right now currently, some of the times it's like generalized medicine. It's like one size fits all, where whenever you have a specific condition, you give a specific treatment. But we are moving towards a world of personalized medicine, where we have to have diagnostic tests in terms of biomarker analysis or lifestyle analysis or diagnostic tests specific to each patient, and based on what a combination of these tests tell you, you have to decide what treatment to give rather than one size fits all. And that would probably reduce your amount of non-responders or patients with serious adverse events to your treatment. Now, the time has come that we do identify the non-responders, and probably will need advanced diagnostic modalities for a customized approach. So what do we need? Probably it's a biomarker. A biomarker is nothing but any imaging or molecular biomarker which can be found in your blood, other body fluids or tissues that is a sign of normal or abnormal process or a condition or disease. And the biomarkers can help to predict the disease. It can help to see whether this is safe or not. It can help to diagnose the disease. It can help to monitor whether your treatment is working or not. And it will also tell you whether this specific biomarker has a potential or a very high risk to a certain condition or a certain eye as well. So when we started preserving all the samples which we technically remove and discard during the surgery, like tears during your dry eye test, blood serum, vitreous, lenticules during your smile surgeries, aqueous during your cataract surgeries, and epithelium during your PR-Cure cross-linking surgeries. We started collecting all those samples in this Eppendorf tube and started storing them in our repository. And whenever we wanted to find an answer to a particular question, we used to remove them and do probably a molecular factor analysis using a flow cytometry or a qPCR to find the answers. But we all know that it's quite every clinician cannot have it in the clinic, so we wanted to develop something which every clinician can use in the clinic, and that's why we have developed this BioM Pathfinder kit, where in your clinic, along with your Schirmer strip or aqueous or vitreous, you can measure 8 biomarkers at this point of time, which is an ELISA-based kit, where it's a microfluidic device where you just add the samples in the cartridge, and at the end of 90 minutes, you get a report. So just a demo of how the test is done. This is for tears for any ocular surface condition. We collect a Schirmer's tube at the end of 5 minutes, like how we do a dry eye test. We add a patented buffer solution in the tube and then shake it for 5 minutes for the protein to be eluted. And then at the end of 5 minutes, we take 5 microliters of the solution, put it in the cartridge, and put it in this device. At the end of 90 minutes, we get this detailed reports, and our journey has now come to an AI-based report, where based on a combination of these 8 biomarkers, for each of the right and left eye, we will come to know whether it's healthy, whether there is subclinical inflammation or inflammation present. And this is based on AI, so it takes into consideration the combination of all these 8 biomarkers. Now, what biomarkers we have chosen? We have chosen a combination of inflammatory and non-inflammatory biomarkers, and we have also chosen biomarkers which can be used for your dry eye patients, your keratoconus patients, if you are a retina surgeon, it can help in your ARMD and diabetic retinopathy or vascular occlusion treatments, and it can also help in your ROP babies as well. So that's why this combination of treatment, so that this kit can be used as a multi-purpose kit by an entire institution or clinic. And as I mentioned, that the report does tells us whether this is normal or abnormal. How does it help us in clinics? We do have a variety of biomarkers. So if there is only one biomarker which is on the higher side, based on what levels, whether it's 1-fold high, 2-fold high, or 3-fold high, you can customize and titrate the treatment based on the severity. If it's ICAM1, you can add a lifitegrast to the patient's treatment at the first line of management so that there are lesser incidents of non-responders. If it's a combination of biomarkers, then you can think of an IPL or a vector pulsation therapy along with lubricants and try to avoid steroids as much as possible to avoid unnecessary complications. And this is what we do for our dry eye disease patients and keratoconus patients. ROP babies, it's quite tough to see these babies, what they suffer, especially the premature babies, and sometimes you do have to wait to see whether the disease is going to progress or not. So taking a step further, we use this biomarker kit to predict that whether a particular ROP baby is going to progress or not at the first visit itself, and we titrate the treatment, whether that will need an anti-VEGF or not, and whether the baby will need a laser, and that's how it helps us in ROP as well, based on the VEGF values. Now, as mentioned before, this kit is not just for dry eye or keratoconus, but it can be used for many other unhappy patients, vascular occlusions, retinal conditions, myopia, and glaucoma, and that is something what is our aim. The future would be that the myopia is on rise, and we do know that sometimes we cannot predict whether which patient is going to have a progressive myopia or which patient is going to have a non-progressive myopia, and whether your treatment is going to work or not. So a very important biomarker which is associated with the progression of myopia is dopamine. If your dopamine levels are on the lower side, these patients are more prone to have mild or moderate or high myopia. And if it's on the lower side, it can keep increasing as well. So the dopamine level here is the key, and it will also help us to identify the customized treatment or frame a treatment for these patients based on the dopamine levels and what we do and what best drug would be there. It also, based on the tau protein, it also helps us to predict whether a particular Alzheimer's patient, what is going to be the path of it, whether a particular patient is going to develop Alzheimer's or not, and if yes, then how fast is going to progress is what this kit can tell us and help us to predict. Now, when it comes to keratoconus or any post-refractive surgery ectasia case, we don't know that why some patients stay stable and some patients do progress over time. So we are moving towards an era of imaging as well, and in keratoconus the key is in collagen imaging, but at this point of time the collagen imaging can only be done when we extract the corneal tissue and do some gene expression analysis. But is there a non-invasive way? Yes, the non-invasive way would be polarization sensitive OCT, which helps us to measure the collagen orientation and the Bowman's layer thickness, like this map. If you see here, the darker the blue color or violet the color in the center, that means your density of the collagen is quite good for this particular patient. And if you see certain patterns, like how the collagen fibril orientation is, then you know that this is a healthy eye. But if this is disrupted, then you see more yellow or red colors in the periphery. And it also gives you, the PSOCT also gives you the Bowman's layer thickness, a thick Bowman's membrane which is above 15 microns. That means this patient has a less risk of developing ectasia or going into ectasia post-refractive surgery. However, if it's a patchy Bowman's layer, like you see an area of thick Bowman's layer and a thin Bowman's layer, this patient, if you have to perform a refractive surgery on this patient, then this patient is at a high risk of developing ectasia. So PSOCT will help in doing this because it can come in a permutation and combination of that, and based on that you can decide the surgery for your refractive surgery patients. So we are moving towards an era of personalized medicine. All of us do try to give the best way where we can give a personalized approach based on biomarkers or imaging modalities, and I think we just have to enhance our diagnostic modalities as we go on with time to give a personalized approach. Thank you.
Thank you very much. I think we can have time for questions. One question. Do you have any questions? Yeah.
Sir, at this point of time, as soon as we get the approvals, hopefully in January 2026, it will be commercially available. January 2026. For now, yes, sir, because we have an in-house kit available, so we can do the test and give the report. Thank you.
Okay, thank you very much. Thank you, yeah. Our next speaker is Professor Namata Sharma. Yeah, I think she's the scientific organizer of the conference, so maybe, I think she's busy in other sections, so she's not here. So maybe we can move on to the next one. The next one actually is me, so I'm going to go there. Yes, okay, yeah. So, yep, thank you very much. I'm Wai-Kit Chu from Hong Kong. So I'm going to talk about keratitis today. So we're trying to do some translational work based on our laboratory finding. Because I'm a laboratory scientist, I'm not ophthalmologist. So in terms of lab work, actually, I work on cornea, obviously. I also work on other eye diseases, like on retina, glaucoma, uveitis, retinoblastoma, because I'm the scientist in the department. And my work is to translate our finding to help patients. So today I select this one to fit in our theme of this section is the translational work. So this is a work based, I mainly work with my previous PhD student, Binning. Now she has her own group in China, in Qingdao. So later I will talk more about that. So this one is actually like, I have no conflict of interest to declare, and try to talk about this bacterial infection, Pseudomonas aeruginosa. This is a bacteria like gram-negative. It is an aerobic bacteria, wart-shaped, and it is highly virulent. So now actually we are using antibiotics to treat it, especially if there's infection in the cornea, ocular surface. But now we also try to explore any new antibiotics or a new method to kill this bacteria or to treat the disease. So this talk is mainly talk about our finding a new pathway actually affecting our body more than on the bacteria. So and we try to work on the contact lens wear, especially in lab animals, because we found that on mice, if you just inoculate some bacteria, the result can be fluctuating. Sometimes you got the infection, sometimes you don't. But if we also apply contact lens, which is mimicking the human situation, like most of the contact lens user, they have the scratching or ocular surface, a little bit of damage that will make them easier to get infection. So we use this as a model to study this bacterial infection. And also these contact lens wear, we know that in human data, this is a major risk factor because there are multiple mechanisms, as I said, mechanical injuries, also hypoxia, the low oxygen level on the cornea may help some bacteria to grow as well. Also that would affect the tear composition. Also is the microbial composition, because our data after this, we also found the contact lens may help or encourage, at the same time inhibit certain species of the bacteria to grow on the cornea and the contact lens. Yeah. And this is our earlier paper a few years back. Actually, we tried to study a neurotransmitter, norepinephrine, NE level, if we put on the contact lens on the mice. So we actually found if we put on the lens, on the ocular surface, we can measure high level of this NE concentration after we put on the contact lens. This one is actually, we use an animal model, we put on the lens first, and then we do an eyelid suture to half close the eyelid to hold the contact lens in place. And then we measure the NE content. Also that at the same time that if we take out the contact lens after, we can measure the bacteria, this aeruginosa has higher concentration on the contact lens. And then in figure B, you can see that we use the electron microscope to look at the control and the NE, we apply additional NE, and that can help the growth of this bacteria on the contact lens. And then this is we work on the animal model now. We inoculate the bacteria and then apply a contact lens there. At the same time, we can add the NE and observe any changes on the ocular surface. You see that if we apply the NE after 72 hours, we see more infection, keratitis compared to the control without NE application. And then in terms of clinical scores, also the level of bacteria also, we can see more bacteria on it. And then the bacteria on the cornea, not only on the cornea, but also on the contact lens, we see high level of this bacteria growing there. So, and also then we try to do the histology, look at the immune response on the cornea. So we see that it's the infiltration of the neutrophil into the full thickness of the cornea. So you see, like if we add the NE, that actually the whole thickness, we can see this neutrophil marker, as well as other cytokines like TNF-alpha, IL-1, IL-6. All these are pro-inflammatory cytokines. We see higher concentration. And at the same time that we can use a drug to deplete the NE, this DSP4 is a drug. So the control now is that we, with the NE, but at the same time, if we add this DSP4, we can suppress deplete the NE and suppress the infection, also in clinical score and the bacterial load as well. And also the bacteria on this ocular surface, or on the contact lens, we see the lower bacteria here. So this, if we deplete the NE, that seems is useful. Hopefully we can translate that as a treatment. And then after that, we follow that with another paper. We try to study more about the mechanism of this infection, NE-mediated infection. So we know that, like in the cells, there are different subtypes of this NE receptor. It's called the ADRB receptor. And we found that we try to look at the corneal tissue. We found this one subtype is the ADRB2 receptor is highly expressed on the corneal epithelial layer. And then now we can generate a heterozygous knockout mice or knockdown mice with only one allele of this ADRB2 deleted. And then we have the wild type and control, and at the same time we apply the NE and inoculate the bacteria. And now we see that only in these plus minus mice we see a lower bacterial level in the cornea. And then now we try to study in addition to use the knockout, genetic knockout, we also try to look at the drug, this receptor ADRB2 antagonist, or ICI, or the ISO is the agonist to stimulate this receptor. We found that only if we use actually here, like if we use this ICI, the antagonist, we can have lower level of this infection. And then if we look at the mechanism, the ICI actually would induce expression of this, they call the double specific phosphatase, DUSP5 is induced expression. So and to prove this downstream pathway of this receptor, we have a drug, is the suramin. If we apply co-treatment with the ICI, now we see the infections coming back to confirm this DUSP5 and the and this ADRB receptor, they are in the same pathway. And then later, we try to study what is the effect of this NE receptor ADRB2. So we try that, we apply this ICI, and then we look at the corneal, between the cell, the junction protein, the Z01. We found that actually if we use this antagonist to suppress the receptor, there are less space between the cells, like as you see in the staining, and this is a schematic diagram. At the same time, if you stimulate this receptor with the agonist ISO, you see now on the staining, there are a lot of gaps between the epithelial cells. So actually, we believe this is generate some gaps for the bacteria to infiltrate into the cornea. So that means that actually this is the pathway that we are seeing is working on the cornea, on the epithelial cells, instead of inhibiting the bacteria. Of course, now we are also studying the impact of the bacteria on the bacteria as well. So now, the conclusion here is that we found this NE pathway and also the receptor that can promote the contact lens wear induced keratitis. I think this is the end. And as I said, this is my previous PhD student. Now she's working in China with her own team, and this is my current team in Hong Kong. So they are all working on different eye diseases. That's all. Thank you very much. Thank you. Any questions? Okay, so if not, then I go back to my seat. So, yeah. Okay, thank you very much. And then our next speaker is Professor Mehta. Yes. Professor Mehta is going to talk about drug delivery system for allergic eye disease. Thank you very much.
Thanks. Slides.
No connected.
So I'd like to thank the organisers for the invitation to this symposium again. So keeping on the theme of translational research, I'm going to talk about drug delivery systems for allergic eye disease. These are my disclosures. So all of you will know that basically allergic eye disease is a continuum that affects a lot of our patients, almost up 5 to 30% of our population itself. And you'll all be aware of the symptoms that our patients have with itchiness, redness, and foreign body sensation and tearing and photophobia. And if it's very bad, certainly in children, it can certainly affect their quality of life. There's a whole spectrum of allergic eye disease, and VKC, or vernal keratoconjunctivitis, conjunctivitis, is probably one of the most severe forms, and there's different phenotypes of VKC, depending on whether it's either mixed, limbal, or basically palpable as well. In Asia, we may see more of the mixed type as opposed to the limbal phenotype, which is a bit more common in European countries as well. And one of the things that's really changed in the management of EKC over the years has been a paradigm shift in basically where we introduce immunomodulated therapy in the treatment. This paper from here that was done by the Movia study group in Asia basically shows you how there's been a shift into basically starting immunomodulated therapy, such as basically with cyclosporine, earlier in the treatment before actually even starting topical steroids. The paradigm shift really came about from these two publications, basically the VECTOR study, and it was the introduction of low-dose cyclosporine by Santen to this cationic formulation using 0.1% cyclosporine dosing that really has basically shown that we should really think about starting cyclosporine therapy early in our patients, and actually even if you basically have to give steroid therapy for any breakouts with respect to an increase in inflammation, you can actually cut down the duration that they actually need to use topical steroids, and of course all the side effects that go along with topical steroids as well. So clearly there's basically a bit of an unmet need itself in any basically topical formulation itself, because there are actually very few formulations that are basically available. One of the other things is that actually we know that actually tacrolimus is a far superior drug to basically cyclosporine because it has almost 50 times amount of efficacy itself because of its mechanism of action itself. But again, there are very few commercial available preparations. Eye drops themselves, of course, they're in a suspension formulation, so they can cause blurring of vision. There's always an issue basically with compliance itself, and especially if you use basically tacrolimus, this can cause a lot of irritation itself. And often if you do have to use tacrolimus, most of us basically will use Protoptic as a skin cream and use it onto the eyelid, which is an off-label use. It's very difficult to get commercialized formulations. Senju Pharmaceuticals in Japan do it, but otherwise it's actually quite difficult to get it. The other option basically is to try to compound it, and there can be a large variability in the dosing itself. So there's several hurdles with basically the use of basically standard of care, which is obviously cyclosporine. It can be a slow onset of effect, poor bioavailability, barriers to basically patient compliance, and of course this can have a significant effect basically in the child's quality of life, and also with the parents, because of course the fact that the parents have to administer the eye drops for the children as well. So one of the ways of getting around this is to think about using a nanotechnology-based system to basically put it as a drug delivery. And if you're looking at nanotechnologies, there's several different formulations like you can see from this schematic over here, from nanoemulsions all the way down to nanoparticles and lipid nanoparticles as well. And we're going to be talking about nanoliposomes, which is the technology that we developed in basically Singapore itself. One thing to understand is that basically you have a choice of basically doing cyclosporine or tacrolimus. And the reason why basically we've chosen to go with basically tacrolimus is the fact that we know tacrolimus, even though it has a similar mechanism of action, is way more potent than cyclosporine itself. And the limitations have been the lipophilic structure of tacrolimus, and that's why we don't have an eye drop basically available. And that's why most of the companies have gone with basically cyclosporine itself. But we thought if we're going to make a drug delivery system from scratch, let's go with basically the stronger drug, where we know there's basically a lot of efficacy from there. So we started basically in this concept a few years ago. So this is one of our publications back in 2018. And the concept was basically put this onto a sustainable release microfilm, where you can see in this animal model that we used, that we're able to get pretty good sustainable release above basically therapeutic concentrations. This is basically using in vitro data and comparing it to basically in vivo studies. And this is using the allergic eye disease model and basically show an effective outcome with respect to release profile. But it only lasted basically about a month when we basically put it into a film liposome itself. And we thought in a practical circumstances, this is not going to really be very clinically translatable as well. So we switched then basically to using a nanoliposomal formulation itself. And this is basically the basis of the formulation we use. So it's basically made of cholesterol, so POPC, basically synthetic lipid base. It's because the tacrolimus is very lipophilic, so it basically goes into the coating over here itself. And on initial in vitro studies and also in vivo release studies in rabbits, we were able to basically get about a 3-month basically release profile. So we thought, okay, this is quite reasonable. We could basically have a 3-month release from one subconjunctival injection of the lipotac as well. And we also thought administration was a little bit easier as well. So this is the manufacturing process of basically how you can actually manufacture this liposomal basically formulation. Initially, obviously, most of the studies were done in-house in the engineering university in Singapore and NTU. And now you'll see later on when we go to commercial clinical trial, we get it formulated in this company called Formulatica, which is based out of Australia as well. So the in vitro studies on the left-hand side, basically it targeted that this is the release profile that you get. So you get initial burst and you get a concentration or release profile up to 100%, up to about maybe 6 to 8 weeks. This is doing in vitro basically testing. And when this is tested above therapeutic concentration, which is equivalent to what you get with an eye drop, what you can see below with this line over here itself, you can see you can maintain well above basically eye drop basically topical concentration of tacrolimus, well up to basically about 7 or 8 weeks itself, above basically the target dose. We stopped the experiments basically over here itself. If you inject this into a rabbit eye and then basically you retrieve, you can see the difference in the concentration on the ocular surface in the rabbit when you basically take the rabbit conjunctiva and then basically look at the concentration of basically tacrolimus. These ones in the brown bars over here itself is the liposomal injection. The one in the green is basically the topical therapy. So you can see the variation you get in basically topical therapy dosing compared to the sustained release that you get with the liposomal concentration. We then basically went back to the same efficacy model that we basically used before. So this is basically using a rodent model. This is a mouse model that's used a lot for basically animal studies. And you can see from the bar chart over here itself, compared to blank control, we're able to show a good efficacy with respect to reducing the inflammatory response in this basically animal disease model of inflammation. If you take basically the corneas out and you basically look at inflammatory cytokine markers, either using immunohistochemistry on the left-hand side or qPCR, we looked at basically IgE-mediated inflammatory responses through typical interleukins 4, 5, 13, and CD40L. These were all basically reduced by over the 4-week time period following basically the lipotac formulation itself. So with the animal data, we did basically the efficacy data. We then thought, okay, now it's time to basically think about basically taking this to a human clinical trial. This is the actual formulation that we basically use for the patients. So this is actually what's actually in the actual vial that you basically get. So you can see the dosing of basically tacrolimus, water basically for injection itself. This can be stored in a normal refrigerator and then just basically taken up into a syringe. We then basically prepared a dossier, as you have to do for any translational research. We had to do GMP batch studies, which were basically coming from Australia. The GLP toxicology studies were actually done in India. We then had to do pharmacological studies for PK/PD release profiles, then made a dossier, submitted this to basically HSA for clinical trials application, and obviously got basically an IRB. So the whole of this process took the best part of almost 1.5 to 2 years to basically get done. This is the release criteria for the tacrolimus before it's basically sent to the physician. You have to basically look at the quality of the product itself, the pH, the stability, and also obviously has to be sterile with respect to endotoxin basically use as well. So this is what basically comes in the vial. And this is our basically developmental timeline. Now we're basically up to basically just completed the phase 1 study in Singapore. This is how you get it in the clinic. So it comes in a little vial like this. I'm just transferring it to the girls take it out of the fridge. I'm just transferring it to basically a 1 ml syringe over here. So it's done basically in a clean room. So you don't need to do this in OR. You can do this basically either in a treatment room or basically you can do this on a slit lamp over here on this side. This is one of the boys who has VKC. So these, in the first-in-man trials, we took children that basically were controlled with VKC, not on any steroid therapy, but basically were just using cyclosporine. Then we stopped the cyclosporine and we gave them basically tacrolimus, and they have a whole battery of tests, including tear film cytokine analysis and also blood tests to look at the tacrolimus level inside the blood as well to see what the absorption effect is as well. So we've just completed the first-in-man study at the end of last month, and now some of these children are basically following up over the 3 to 4 month period to see if they get any flare-ups of their VKC as well. So the main advantage of this drug delivery system is it's got superior effectiveness and bioavailability. You have sustained release, so you have much better control of the VKC, and a simple subconjunctival injection will last, we think, basically 3 to 4 months without having the patient basically having to come back, and there's obviously going to improve compliance. It's safe. So far, we've had no issues basically with the injection, and there's no issues in systemic absorption or basically tacrolimus. And of course, potentially, now we've basically done this as a disease model for VKC, you could potentially use this for other diseases such as graft versus host disease, Stevens-Johnson syndrome, and also MMP as well. To conclusion, allergic eye disease is a common modality. We all basically see this in clinic, and of course, it's increasing in incidence. The spectrum basically with VKC is on the severe side, but they have very defined phenotypes, and that's why we chose this to do our first-in-man basically studies. Obviously, it's particularly common in children as well. There is a paradigm shift on VKC treatment to using less steroid use and to use early use of immunotherapy, typically with cyclosporine. However, obviously, there's issues in formulation availability, especially with basically tacrolimus. Preparation and compliance is a huge issue, and we think Lipotac will basically solve all of these problems, and our first-in-man study so far have been encouraging, and hopefully we will publish this over the next year. Thank you for your attention.
Thank you very much. Now we have time for one question. Do you have any questions for Professor Mehta? Oh, just a quick one. Like
Yeah.
in your phase 1 trial,
Yeah.
how many patients you
6.
tested? 6, only 6.
Yeah, units. Phase 1 normally basically is 6. It's single dosing,
Yeah.
and it's not repeated
And
dose.
it's only one eye.
It's only one eye. So the other, so what we did was these kids are all. So the problem is that with these kids, they have to be on cyclosporine, completely stable, never taken steroids for basically over 2 years. So they're only on. And then what I did was I stopped the cyclosporine for both eyes, and then we basically have been monitoring them. So actually, that kid was one of the first ones that we did. So he had, I think I had it in January and stuff. So actually, his other eye now had a bit of a flare up because I stopped the cyclosporine. So now I've restarted him with cyclosporine on the opposite eye. But so far, the eye that had the tacrolimus, he's been absolutely quiet. In an equivalent formulation of they didn't call it Lipotac, in an equivalent liposomal formulation for Xalatan for glaucoma, when they did their clinical studies, they found that in patients they had stability or effect for almost 4 to 6 months. So you may actually get longer duration of effect itself, but this is basically what we were looking basically around about 3 months. That's what the animal studies for, and that's exactly what they show with Xalatan, but clinically they were able to get longer itself.
Okay. Thank you very much.
Thanks.
Yes. So now our next speaker, actually our previous speaker, now she's with us now, like Professor Namrata Sharma. She's going to talk about the Natazole enhanced drug delivery in fungal keratitis. Thank you.
आगे So I'll be talking to you about Natazole, the enhanced drug delivery in fungal keratitis, and there are no financial disclosures. And when you use topical natamycin 5% in a case of fusarium keratitis, this is how the response should be, but it is not always like this. And basically 5% natamycin is the mainstay for any case of fungal keratitis, but there are limitations in the form of commercially available preparations, poor ocular penetration, poor bioavailability and toxicity. So the indications, we sometimes do the targeted drug delivery in these cases, which includes intracameral antifungals, such as this, which causes the resolution of the ulcer. And sometimes also the intrastromal injections are required because the penetration of the natamycin is not that great. We did publish the TST protocol, or the topical systemic targeted therapy protocol in fungal keratitis. And if the fungal keratitis is not responding for 4 weeks, then one can give either intrastromal, intracameral injections, or both, depending upon where the exudates are. Now, what we've been working on is natamycin 5%. Natamycin is water soluble, insoluble, has 5% suspension. So also because it is insoluble, it causes local irritation and excessive tear secretion. And tear dilution also leads to the drug loss. Suspension formulation should be mixed properly before instillation. Natazole or natamycin 1% is a soluble form of natamycin, and And it is characterized, basically characterization of the complex natamycin is done and is performed by the Fourier transform infrared spectroscopy and differential scanning calorimetry. And it is tested for stability in conditions related to patient use in white light at room temperature, dark room at 25 degrees centigrade, and by opening and closing the vial once a day at 4 degrees centigrade. So we did single and multi-dose transcorneal permeation studies of Natasol 1%, which was soluble form, versus 5%, which was suspension in albino rabbits. And sterile, unpreserved, ready-to-use water-soluble natamycin for intrastromal injection was developed using the ARVO guidelines. It is protected against the degradation under various stability circumstances. This is what we found out. And also, it prevents natamycin photodegradation as well. This Natasol 1% we used as topical drops and also used as intrastromal formulation. And we have patented this drug. So just looking at the FT infrared spectroscopy, this is for natamycin and this is for polymer. So when you combine natamycin with polymer, this is what you get. And again, if you look at the representative scanning electron microscopy, the polymer looks something like this, and the pure drug is like this. And the combined, when it gets when you have the Natasol complex, then this is what is obtained. So in terms of drops, Natasol is like clear solution, slightly yellowish in color, but natamide or suspension is not a clear solution, but it is a suspension. And I would like to acknowledge Dr. Vel Pandyan from Ocular Pharmacy who works on it. So we did single-dose transcorneal permeation studies and multi-dose kinetics. The single-dose kinetics did show, if we compare Natasol with natamycin, that the 1 hour permeation was higher as compared to the suspension. And again, this plateaus, but at all points in time, it is higher than the natamycin suspension when we compare it with the natamycin suspension. In steady state pharmacokinetics after hourly and 2 hourly dosing of multi-dose kinetics also confirmed the same. So this has been now put into small tubes, and basically what we require is we have natamycin and cyclodextrin mixed in water. The supernatant is separated and lyophilized, and required amount is mixed in water and sterilized by 0.22 microns filter. And the final formulation is subjected to bacteriological and fungal culture tests before use, and the final sterile drug with osmolarity of 290 milliosmoles was used. The formulation was dispensed in ampules such as this, and we evaluated the role of Natasol in 60 patients. Group 1 had 20. Each of them had 20 patients. In one group, we put only natamycin. In the other group, topical natamycin and voriconazole. And the third group, topical Natasol. And the follow-up that was done was at day 3, 1 week, 1 month, 2 months, and 3 months. So if you see the visual acuity, in terms of visual acuity, there was no difference found between the 3 groups. In terms of the infiltrate size, it was comparable between the groups. There was a significant difference in the rate of healing between the 3 groups, and the patients where Natasol was used was found to epithelize much faster. And if you look at the anterior segment OCT pictures of those also, we found that the Natasol had treated, this is the Natasol treated group, where the deeper ulcers could also be treated. This is the combined group, and this is topical natamycin, and these are just representative pictures. So we did publish the results of the same, that is the surgical outcomes with intrastromal natamycin, which was also used. Mean age of the patient was 40 years, and organism isolated was Aspergillus and Fusarium both. And the cure rate was very high, to the tune of 95%, which is not expected in cases of fungal keratitis. Subsequently, we also did a randomized control study where we compared the intrastromal injections of voriconazole, amphotericin B, and natamycin in cases of recalcitrant fungal keratitis, and 60 eyes of 60 patients were used for this. And again, in one group we gave intrastromal voriconazole, in the other group intrastromal amphotericin B, and the third group intrastromal natamycin. But the adjuvant therapy was continued in all of these groups. Again, these were evaluated in terms of the baseline parameters. All of them were well matched, and the fungal organisms were also, there was no difference between the 3 groups. and hypopion also height was matched. There was a reduction in height of the hypopion, and there was no difference in the 3 groups. Repeat intrastromal injections were given in all the 3 groups, and again, this was not found to be statistically significant. The height of the hypopion decreased significantly in all the 3 groups, and this is one case of intrastromal natamycin, day 1, and this is how it resolved at day 14. Then the reduction in the height of hypopion was significantly better in cases of natasol as compared to voriconazole. And again, we found that the percentage of high eyes or the response was found to be similar with natasol, where natasol was used. The scar size, we did not find any difference in the 3 groups, and also did not find any difference in the 3 groups in terms of vascularization, whether it was superficial or deep, or in terms of the quadrants of vascularization. What we found was that the amphotericin B group was the group where vascularization was significantly more as compared to the natasol group and as compared to the combined therapy. Sorry. So this is another case of intrastromal voriconazole which was given. Notice here the lesion is in the posterior part, but at 6 months it does heal. This is another case where intrastromal voriconazole was used in the same cohort, and this is where intrastromal amphotericin B was used in the same cohort. This is again a case where intrastromal natasol was used, and yet another one where it was used at 6 months. This is again, these are again the cases from the same cohort where intrastromal natasol, that means the soluble form of natamycin, was used. This was fusarium keratitis. Again, this resolved on therapy with intrastromal natasol. So this is just to show the representative picture. So as far as the healing is concerned, it was fastest when we used the natasol or systemic or soluble natamycin. And in those cases where amphotericin B was used, there was marked vascularization as compared to the other 2 groups. So we conclude that novice natamycin inclusion complex enabled formulation holds a promising role and useful adjunct to standard therapy in management of recalcitrant fungal keratitis, which means that it can only complement it, you cannot use it standalone. Then regional differences in microbiological profile should be considered before prescribing them, and we do require a longer term follow-up and larger numbers to validate the current results. This is our publication in Survey of Ophthalmology, and this is in Ocular Surface. I would like to acknowledge the people who worked on these 2 studies, and thank you very much for your kind attention.
Thank you very much, Professor Sharma. Any questions? Yeah.
So, unfortunately, the antifungal drugs are under rate contract, and so they do not, the companies cannot make too much of revenue out of it. We talked to 2 companies who refused, but there's a 3rd one. I think the 3rd company should be willing to take it.
Okay. I have a question about the formulation. So you just mix with the simple mixing the natamycin with the polymer, or there's a chemical reaction to link up the molecules?
I think there is a chemical reaction to link up both of the polymer as well as natamycin, and this is done in our ocular pharmacy lab. So I wouldn't know the details by our ocular pharmacologist, but it is not simple mixing.
Okay, yeah. Thank you. Yeah, beautiful work. Thank you very much. So now we can move on to our next speaker, like Dr. Julia Chen, also my colleague from Hong Kong. She's going to talk about topic dry eye disease, translating scientific discoveries to practice. Thank you, Julia Chen.
Thank you for the kind introduction. I'm Julia Chen from Hong Kong, as the chairman has introduced. And as a new faculty member joining the department, basically, majority of my work are based on clinical works. And I would like to carry on with my talk with a more clinical approach. I have nothing to disclose financially. So dry eye disease, as mentioned by our previous speakers, is a very serious problem. The global prevalence is occurring in rating to even like 30% of the population. In Hong Kong, the prevalence is around 7.7 to 30%. The current model of diagnosis is thinking that dry eye disease can be classified into aqueous deficient, evaporative, or mixed kind of disease. Why dry eye disease is a problem clinically is because of the ambiguity in nature in terms of dry eye disease. As illustrated in the OSDI, the symptoms of these kinds of patients are often very ambiguous. They cause sensitivity to light, grittiness, irritation, which mimics a lot of very serious and sinister condition. And often it can be misdiagnosed by many doctors and eventually causing patients dissatisfaction. The challenges of dealing with dry eye disease is that the patient's expectation is often mismatched. Because patients are so bothered by those irritating symptoms, they perceive themselves having a sick role. They thought to themselves that they have something very serious, but when ophthalmologists disregard their symptoms and just prescribe them with lubricants, they are often unhappy and unsatisfied. So this is a few pictures of the lubricants commercials in Hong Kong, illustrating how common this problem is and how big the market is because of the dry eye disease not being able to be treated adequately in our clinical settings. So what are the symptoms in dry eye disease? It causes punctate epithelial erosion, more often seen in patients with aqueous deficiency, or the patients with meibomian gland dysfunction, illustrated with telangiectasia over the meibomian glands. And when we try to express the glands, the meibum expressed are often like toothpaste and very mucoid in nature. So in order to cater the problem of the mismatched management time and patients' report, and we have established a tertiary clinic solely dedicated to dry eye disease. It's a combination of nurse-led clinic as well as a corneal team, which we combinedly assess the patient. The aim is that we better diagnose patients with underlying pathophysiology, causing better patients' report. We follow up the patients and try to triage the patients into suitable treatments. So the comprehensive approach starts with nursing assessments, doctor's assessments, finally with provision of treatment. Usually in a clinic's time, the nurse will take an hour to take a very detailed history, performing all the dry eye-related tests and questionnaires for the patient, for 3 particular patients, and then these patients will be slipped into our doctor's clinic as a very few last cases where we can adequately assess those patients. So this is the assessment form we have performed. You can see that the nursing staff take a lot of effort in filling in all the forms with taking a very detailed history in a very standardized format. And then they also help us to perform all the tear breakup time, Schirmer's test, sleep lamp examination, try to express the glands. They had a lipid view report and also keratography 5M reports as well. And as last, the nurse will try to suggest some non-medical treatment for the patients. So what are the results with our clinic, which has been operating for already 4 years? So through this dry eye clinic, we have a cumulative of 151 patients with an average age of 61, with female predominance. And you can see that these kinds of patients often are primary dry eye patients without underlying disease, such as Sjogren disorder or secondary Sjogren related to rheumatoid arthritis or SLE. So one of the very significant findings we find in these kinds of patients is that when we trace back the records, we find that it's a highly significant, higher proportion of patients with high ANA theta. So on the left is literature quoting the healthy populations. You can see that in healthy populations, there's still around 15%, around 10% of patients with an ANA theta of about 1 to 160. But then, in these primary dry eye patients, they don't have any prior Sjogren syndrome. They don't have secondary Sjogren syndrome. Nevertheless, there's a high increased patients with high ANA theta level. And fundamentally, these patients are different from those with low ANA thetas. They have low tear breakup time, and their Sherman tests are significantly different. However, in terms of other parameters such as OSDI, conjunctival staining, redness, tear film, and also percentage of incomplete blinking, and also redness, there's no significant difference. So how does this result help us to provide a better comprehensive treatment for the patients? So before anything, we start with some things with basics. We tell the patients education pamphlets are distributed to them. Detailed explanation of how ocular surface lid mark hygiene can be performed for them. Of course, we start with lubricants, and some of the patients who need immunomodulation will provide them. And then we also offer treatments to them. So how do we also did a retrospective analysis, finding that especially in our Chinese cohort, after a session of thermal pulsation system, namely lipid flow, there's significant improvement in these kinds of parameters. Since, as I have mentioned, patients with high ANA thetas, they are fundamentally having a lower tear breakup time. So that's why these kinds of treatment will benefit them significantly. So in conclusion, anti-nuclear antibody in dry eye disease may pave a fundamental for many basic science research that maybe I can collaborate with the chairman in the future. And this illustrates how we can translate our clinical findings into future scientific analysis. Thank you.
Yeah, thank you very much, Dr. Julia Chen. Any questions for Julia? I have one question. I want to ask about the ANA test. So
yes.
Now that is a standard test in your clinic, so most of the primary dry eye, you would send them for this test?
Yes, I will do that for them, yes. And someone will have asked, why do I use ANA as a biomarker, but not other tests? Because when reviewing these patients, I find that there's no other raised titers in these kinds of particular primary dry eye patients. For example, namely double-strand DNA and other kinds of biomarkers, I don't see an increased trend. But there's only just the ANA that is raised in particular primary dry eye patients.
And this test, is it specific to primary dry eye or any other conditions would also show positive?
Okay. In current literature, there's no analysis on dry eye patients on ANA and the relationship of primary dry eye. There are studies previously in Taiwan who looks at the ANA titers in Sjogren syndrome patients, but never on primary dry eye patients. Thank you so much.
Yeah. So is there more questions for Dr. Julia Chen or even other previous speakers? Yeah. So if not, then thank you very much. Yeah, maybe I can say a few words to conclude this section. So thank you very much for all the audience coming here to listen to us. This is a translational section because now in APO we have a subspecialty in focusing on translational research. And this year we want to talk more about the anterior segment. So in this section we have professors talking about the stem cell, corneal stem cells, also the imaging system, and then the drug development. And I myself is more about the pathway and also the dry eye clinic as well. So I hope you learn a lot, and thank you very much, and I hope to see you again in the remaining APO and other sections as well. Thank you very much.